and I will give Philippe the opportunity to start with his presentations. Now we are live, so Philippe, all yours, okay? Thank you so much for everybody here. We have the opportunity to listen to Philippe Marullo for the BioLabForce Institute, and also for Innovan. So thank you very much, and we hear you. Thank you so much.
Okay, thank you. So I hope that you can see my
yes, we are seeing your presentation. Everything is perfect.
Thank you to see Sitevinitech to give me this opportunity to present studies that are not only mine but are also from another PhD student at the ISVV. And the central question today is how to face the drop of acidity that we can meet in wine, and with some new findings from microbiology. So my name is Philippe Marullo. I'm a PhD researcher. I'm a researcher from the BioLabForce company. And I'm working at the ISVV, that is an institute of research here in Bordeaux. So let me check, sorry. Is the first slide. So the climate change, among the different problems that are generated by climate change, there are sugar ethanol content increase, but also a drastic drop of acidity. And due to temperature increase, to the CO2 in atmosphere and hydric stress, but also from different harvesting strategy, this is a big trend since 20 years. There is also impact of phenolic compounds as aroma, but it is not the purpose today. Today I just show you what is the jet prediction from 2100 with a drop of malic acid and tartaric acid in wine. In grape juice, and also an increase of pH unit, of 0.15 pH unit. Okay. And one problematic is the drop of acidity, and this impact many points of vinification, including aromatic consequences, because by changing pH you change the aroma volatility. But also microbiological stability, because you can induce spoilage microorganisms, and also because you impact mostly negatively the ability of aging of wine. And there are a few solutions. You can act as crop management, but you have some particular trade-off, and I'm not at all a specialist of that. You can also add chemicals, including acids, but it's quite expensive. And the question today is, what about the microbiological solution? So during the vinification process, from the grape to the wine, you have the two fermentation, as you know, alcoholic fermentation and malolactic fermentation. And now we will check what are the microbiology level for increasing pH or for decreasing pH. For increasing pH, some people previously suggested to use Saccharomyces spombi that cannot achieve completely the alcoholic fermentation, but that can transform very efficiently malic acid in ethanol. And there are very few industrial applications. The idea in the past was to supplement the malolactic fermentation. And there are also, obviously, the lactic bacteria that can transform malic acid in lactic acid, as well known, and with a very slight reduction of increase of pH of the wine, because malic acid is a diacid, like white lactic acid is a monoacid. In terms of the decrease of pH, what we can see is that Saccharomyces cerevisiae has a low consumption of malic acid during the alcoholic fermentation and a low succinic acid production. And generally, the pH of wine is a bit less than the pH of grape juice, also due to the CO2 produced. There are also other species, yeast species, including Lachantia thermotolerans, that cannot completely achieve the alcoholic fermentation, but that can transform sugars in lactic acid. And this has an interesting impact on pH and on ethanol production, with a decrease of pH and ethanol production. And we will better focus on this yeast for the first time, Lachantia thermotolerans, that, as you can see here, reroute part of glucose in lactate during the glycolysis pathway. Okay, Lachantia thermotolerans was formerly named Fluoromyces thermotolerans and is located here. So I'm just checking for a pointer. I forget to activate it before, sorry. Okay. I hope you can see my pointer. Lachantia thermotolerans is a clade of yeast that is quite divergent from Saccharomyces group, and is minor in grape and wine microbiota consortium, but is systematically present during the vinification process. A PhD work of Anna Halilovic from the University of Adelaide and also at the ISVV screened a wide population of 94 strains of Lachantia thermotolerans isolated in a wine-related environment, but also in wild. And this important work demonstrates that there is a strong variability in pH and lactic acid production for this species, and there is also a strong variability in sugar fermentation efficiency. As you can see here, one of the main trends of Lachantia thermotolerans is to produce important amount of lactic acid, up to 12 grams per liter, and by the way, to reducing the pH with a pH that can vary at the end of the fermentation between 3.8 and 3.2. This is quite important. And this is due, as I told you previously, by a rerouting part of pyruvate in lactate instead of rerouting pyruvate in ethanol. So one of the consequences of this is that there is a reduction of ethanol in the fermentation achieved by Lachantia thermotolerans. In a postdoc carried out with Biola 4 from Anna, she looked out the ability of 18 strains with a narrow selection by changing the SO2 content, the nitrogen content, and the sugar content of grape juice in order to select a strain able to reduce a bit the quantity of the pH of wine at the end of the alcoholic fermentation. According to the amount of SO2, some strains did not implant correctly in the must, and others are quite more robust to alcoholic to SO2 and can achieve the alcoholic fermentation. Not completely, but in part. And so this is just a selection program. So we selected the strains, and we also work to optimize the process. So the very question is which application perimeter? What is the range of acidification and ethanol reduction? Can we use Lachantia thermotolerans in co-inoculation strategy or in sequential inoculation strategy? And what are the limiting parameters, including SO2, temperature, and nitrogen? As you can see here, we fermented Cerisier alone, Cerisier and Lachantia thermotolerans in co-inoculation or in sequential. According to the modality, you don't have the same effect. For example, here you have a temperature and matrix effect. As you can see here in Viognier, you can see that the lactate produced is only 4 grams per liter, while in Cabernet Sauvignon is more than 12 grams per liter, which is amazing. And this could be due to the matrix effect, but also from the temperature of the fermentation. And you have also inoculation type effect. Indeed, here you have a pH of 3.9 in co-inoculation, but you have a pH of 3.3 due to the high amount of lactic acid produced. And it is due, of course, to the type of inoculation. So this idea is that according to the conditions, the Chancellor thermotolerant strain selected can produce between 1 and 12 grams per liter of lactic acid, reduce ethanol from 0.1 to 0.9, and in all the case, we compare wine that no have any more sugar, okay? And that also reduce the wine pH from 0.1 to 0.7 units. A big tip to remind is that 1 gram of lactic acid is more or less 0.1% of ethanol reduction and 0.1 pH unit drop. Okay. However, we have to be very careful because lactic acid production may compromise the malolactic fermentation if the lactic acid produced is more than 2 or 3 grams per liter of sugar. And so this is a main drawback of the use of this species. Okay. Another point is to revisiting the malic acid metabolism in Saccharomyces cerevisiae. And now we are able to select new strains of Saccharomyces cerevisiae that can manage better the acidity of wine. So this is just a teasing slide that shows you what is the distribution of micropercent, which is the part of malic acid consumed by yeast at the end of the polyfermentation by common wine starter. And what we can do using marker-assisted selection, we can produce DEMALIC starters or acidic starters that in some cases produce more than 70% of malic acid consumed, and in other cases they can also produce malic acid. So to do that, we're using quantitative genetics, and quantitative genetics is just a strategy to make a link between a phenotype and a genotype. Okay. And of course, any phenotype we zero in nature is due to environment change, but also to a part of genetics and a part of genetics that can interact with the environment. And so quantitative genetics is quite useful for understanding biology, for finding genes and genetic regions that control some pathway, to understand mechanism of adaptation and plasticity, that is interaction with the environment, but also for optimizing the wine quality by selecting new starters or by adapting some enological practice. And so this is a strategy that we use in the lab. It is called QTL mapping. So this is an experimental design used by my former PhD student, Emilien Pelletier, that is now working at ISB as well. And so the overall strategy is to cross 2 strains of cerevisiae and to get a large amount of progeny. That is, you cross 2 strains and you have a meiotic recombination. And we did that for 4 strains and generating 2 independent crosses. So all these segregants were sequenced by new sequencing genetic techniques in order to build a genetic map able to discriminate the genotype of each parental strains across the genome. Then what we did, we fermented strains in 3 environments, that is 3 great must, Sauvignon Blanc must and Merlot must, and in some cases the Merlot must was not shook instead of shaking. It is useful for mimic micro-oxidation. And we measure the kinetics traits, so that is the ongoing of the alcoholic fermentation by weight loss, and also the end product metabolites, including glycerol, acetic acid, ethanol, malic acid, and basic compounds. Today I will just focus on malic acid metabolism, of course, and as I told you previously, part of the malic acid in grape juice is consumed by yeast through the malo-ethanolic fermentation. This is a well-known phenomenon. And as the quantity of malic acid in wine, in grape juice, is not exactly the same, we define a parameter that is MAC percent, that is the malic acid consumed by the yeast on the total malic acid. Here you can see the segregation, that is the distribution of the phenotypes in the two populations we generated. So this bell-shaped distribution illustrates that few individuals in, for example, the M2, M15 population have a MAC percent, reach a MAC percent of minus 20%. And so this is the range of MAC percent in the two proteins. By using a quite boring strategy of genetics, we linked genetics and phenotypic variation using statistical approach, and we defined some genomic regions that control the phenotype variation. And we localize where are these genomic regions. So we map 22 QTLs. QTLs means quantitative trait loci, so a locus associated with a quantitative trait. And in those regions, you have some genes that control part of your variation. And so 13 QTLs in M215 and 9 QTL in SBGN, whatever. We also demonstrate that most of these QTL fall in this blue region and are quite robust to the environment. That is to say that whatever the quantity of malic acid you have, or whatever the color of your grape juice, these genes and these regions are able to change quite robustly your phenotype. And this is a good news for selection. And then we also validated some genes in SBGN background by deleting one gene using molecular genetic approach, and we validate 6 genes, sorry. And these genes, and this is just a slide for show you that are quite close from the malic acid metabolism. Indeed, MI1, that is the malic enzyme that transforms malic acid in pyruvate, but also PIK2 that transforms pyruvate in oxaloacetate that can be converted in malate, and so on. So we find 5 genes that are quite close related to malic acid. We also discover that one of our strains, SB, has a mosaic genome. That is to say that it's not a pure wine yeast. As you can see, pure wine yeasts are in green in this phylogenetic tree, and SB is quite close to floor yeast. And indeed, almost 50% of the genome of SB is strongly irritated from floor yeast. And we found that 7 of the 9 QTS we found that reduce MAC percent are located in these floor yeast specific regions. So this is fundamental research that show you that by mixing different kind of yeast, change the metabolism of fermenting yeast. And now we illustrate quite rapidly what we can do by selecting strains and breeding them together. So here you have the position of macpercent of the 4 parental strains we have, and we have the segregation of 94 SBGN progeny. So we selected one strain P2 here that has a macpercent of -5%, or a P4 that has a macpercent of 60%. And we did the same for M2F15 by selecting P1 and P3. And then we crossed together P1 and P2 and P3 and P4. So we did exactly what people that select plants and animals are doing generally, routinely, by selecting extreme individuals. We fix the genes that are able to move a phenotype. And then we selected our strains using a marker-assisted selection. That is to say that since we have identified 22 loci that control a trait, we use those loci for selecting progeny. So we get a large amount of progeny, more or less 2,000 segregants of P1, P2, and P3, P4, and we genotype all of them for these 27 B allelic markers that represent 22 QTS. And then we select just individuals that are MAC plus or MAC minus enriched. That is to say that have a lot of alleles of genes that promote the malic acid consumption, or that prevent the malic acid consumption. And the result is shown here. We get 13 clones that have more than 17 of the 20 MAC minus allele, and we have 35 clones that have more than 8 of 11 MAC plus alleles. And as you can see here, the distribution of these extreme strains are located in this Gaussian, orange and yellow Gaussian band. And as you can see here, the quantity of malic acid at the end of the alcoholic fermentation, the same grape juice, is 3.34 with a macpercent of 0%. That is to say, the yeast does not consume, it does not produce malic acid, but can reach 5.66 in DEMALIC strains, or just 1 gram per liter for DEMALIC strains, and 5.66 for acidic strains, with a clear impact of the wine of the pH of wine. And in fact, there is a direct effect of positive correlation of the pH of wine. So, just 2 slides for show you how can we use DEMALIC starters. These DEMALIC starters can be useful for help Enococcus ani to consume more efficiently and more quickly malic acid and produce lactic acid, of course. And we can do that in co-inoculation strategy and in sequential strategy. So this is a sequential strategy in which we have 6 grape juice containing high, medium, and low amount of malic acid in the grape juice. And as you can see here, between the blue curves and, for example, the red curves, you have a gap of 1 gram per liter of malic acid, in some cases 2 grams per liter of malic acid, and it is due to the consumption of malic acid of some strains. Okay, so the blue one is the GN strains, and as you can see here, we select strains that consume more efficiently malic acid than also SB, and you can see also that the decrease of malic acid due to Enococcus ani, because here you can see the malolactic fermentation, of course. The time can be reduced when you are using strains that consume more malic acid. And so you get a faster malolactic fermentation, and there is no MLF rate inhibition. That is to say that even if you consume a large part of malic acid of wine, the inoculation starter can initiate malolactic fermentation without problem. And so what we can do with acidic starters, of course, it's quite easy, is just to ferment with those starters and to reduce the pH of your wine. And as you see here, there is a drastic reduction of wine pH at the end of the cold fermentation. This is a set of acidic starters, and it's quite 0.2 respect to the best strain of the market for the moment, that is GM, or minus 0.6 with respect to SB, that is another extreme strains. So my concluding remarks are those. La chancellor thermotolerance is able to increase acidity and reduce ethanol content in mixed culture with Saccharomyces cerevisiae. And some ADI yeast are available, at least 4 strains in the market. And so it's possible to use them, but it requires a careful vinification management in order to do not overcome the quantity of lactic acid that can be 16 grams per liter of lactic acid. Is terrific in terms of acidity. And but it's quite efficient also because you can reduce a bit the ethanol content. And now in the next future there will be new Saccharomyces cerevisiae strains because by mixing flow and wine genome we rewire part of malic acid metabolism, and we were able to control the MAC % by using marker-assisted selection and breeding programs. And so we are able to create acidic strains that can drop the pH of the wine, at least from 0.2 units, and also to develop DEMALIC strains that can be used for facilitating the malolactic fermentation. And so just to give many thanks to Hanna Halinovic from the Adelaide University in collaboration with Vladimir Jiranek and my colleagues Warren Bertin, Isabelle Masnaf and Marina Belli from ISUV, and also my colleagues Joanna Coulon and Mario Favier from La Four. And for the Saccharomyces cerevisiae project, my 2 PhD students, Emilien Pelletier and Charlotte Vion, and my collaborators in La Four too. So thank you for your attention, and I'm looking for your question.
Hello, Philippe. Many thanks for everything. It was a very professional and clear presentation.
Okay,
Can you hear me? Can you hear me? Is that okay? Okay, that's great. Perfect. Let me ask if we have some doubts or if somebody would like to make some questions. I'm asking here if there's somebody to make a question or something like this. Well, no, apparently there's no doubt. It was really very, very clear and professional and nice, so we really, on behalf of Sitevinitech, we would like to say thank you very much. There's no question, so thank you so much for everything. We know that we have a time difference, so I don't know if you are okay with the time, but here is midday, so thank you very much for staying on the other side. And we look forward to have the opportunity to have another presentation with you in the near future, okay?
Thank you.
Thank you very much. Have a great day. Thank you. Thanks everybody for your presence here. Okay, we will continue with Sitevinitech conferences. Vamos a continuar con las presentaciones. Muchísimas gracias a todos por la presencia aquí. Muchísimas gracias por estar del otro lado. Les vamos a